I hereby open this academic session in which Mirna Anđelić will defend her academic thesis called Neuropathic Pain, Identification of Tissue-Specific Molecular Signatures. Dear Mrs. Anđelić, may I invite you to present us with a summary of your study and its main findings.
Thank you. Good afternoon, and thank you for following my public defense of my PhD thesis entitled Neuropathic Pain, Identification of Tissue-Specific Molecular Signatures. I will take this occasion to tell you a little story about my PhD journey and things that I studied. I believe that every one of us here experienced pain sometimes in our life. But I also believe that not many of you thought how lucky you are when this pain went away. I'm telling you that because there is more than 5% of people worldwide that suffer from pain daily. We call that pain neuropathic pain. It is a pain that this pain is caused by a lesion or disease of somatosensory nervous system, which is a system that is helping us. That is a system that registers what is happening on the surface of our body and sends this message to our brains. Neuropathic pain can be a result of an injury, infection, metabolic disease, toxic exposure, even our genetic background, or it can be idiopathic, which means that we don't know cause of this pain. It is frequently associated with a sentence that is a global healthcare burden. If I consider my background in biomedicine, it was really hard for me to relate to these words, but my supervisor in Carlo Besta Institute, Professor Lauria, knew that, so he helped me to understand it by inviting me to patient visits when that was possible. So in that occasion, I understood how complex this journey will be. Because patients I met there described their pain as burning sensation. Sometimes some patients said that their pain feels more like electric shock-like pain. Some patients said that sometimes it's like burning sensations, other time as electric shock-like pain. Some of them said that explained that they felt presence of exaggerated responses to noxious stimuli, for example, heat, or painless stimuli. For example, it was painful when they would wear socks. When we talked about distribution of their symptoms, majority of them would say that they feel pain that is on their feet, on their palms. Some of them would say that this pain is concentrated on a certain area of their body, or some patients would say that they feel pain everywhere. The only thing I could honestly see that it's common to all these patients is that this pain was long-lasting. And when it lasted more than 3 months, we defined it as chronic neuropathic pain. You may understand that from biological point of view, all these symptoms and signs can be triggered by different mechanisms or can even trigger different mechanisms. So I really didn't know how can I be helpful. But then we came to, then I looked how these patients are diagnosed. In our clinic, we have the patients undergo clinical assessment, specialized pain questionnaires, neurophysiological exams, and different laboratory tests. But we still do not have objective biomarkers for chronic neuropathic pain. And that is a question I thought this is a challenge that I considered very interesting. When patients are diagnosed with neuropathic pain, the medical doctor decides on the treatment, and this treatment, unfortunately, still is directed to treating their symptoms rather than the cause. We do have a couple of drug classes available, among which sodium channel blockers are the new targeted therapy. Sodium channels are channels that are expressed on our nerves that send painful message to our brain. But even when considering this targeted therapy, less than 50% of patients reports pain relief. We do have now available nerve stimulation techniques and non-pharmacological therapies that work well for some patients. In our lab, we are asking ourselves always, how can we help to clinics? So to answer these questions, and as a first question of my thesis, we asked ourselves, what is the contribution of genetic background in neuropathic pain? When we talk about pain and genetics, we must admit that Mendelian or monogenic pain disorders are very rare. But they do exist, and and when they occur, they are very exaggerated. For example, one of the most investigated pain-related gene families are sodium channel genes, and we know that today, thanks to a lot of research that has been done in this field, that they can cause from very extreme painful disorders to even extreme disorder that we call congenital insensitivity to pain, in which patients do not feel pain. I know, it seems like it is something that we all would like to have, right? But I'm here to tell you that actually this disease is very serious, because the patients that because pain and nociceptions are our internal warning signals. So if we would never feel pain, we would never seek help. So you may understand how serious that can be. During my PhD experience, I had the chance to be introduced to one of these very rare extreme cases. A clinical doctor from Turkey sent us clinical data and samples from a little girl that suffered, that had a history of unusual accidents, among which third-degree burns that she got after holding her hand on a burning stove. Combining a couple of molecular approaches, we identified a sodium a mutation in a sodium channel gene that I already previously mentioned. And in order to explain you what really happened to this little girl, I wanted to remind you of the central dogma of molecular biology. If we consider DNA as a master book of recipes that hold all the instructions to make a protein that are building blocks of our body, we can, once our body needs a certain protein, a specific message gets copied, and we call that RNA. And this message goes to a protein-making factory, and the protein gets produced. In this little girl, her DNA contained an error, a mutation, which caused then that this message, RNA, is damaged. So when it arrived in the protein-making factory, it produced a faulty protein that didn't work. In this way, we explained her symptoms. We next looked at pain as a complex disease. So by applying gene burden analysis, we examined the collective impact of genetic changes on the likelihood of developing neuropathic pain. By applying this approach, we identified a gene that was highly enriched with variants in a patient cohort when compared to healthy individuals. This gene in our patient cohort had shared several characteristics, such as pain that was triggered by cold, neuropathic itch, and our patients felt pain everywhere in their bodies. With this work, we identified TRPA1 gene as a risk factor. We then moved to study, to look for these precise biomarkers, molecular biomarkers, for precise patient stratification. Taking a lesson from other complex diseases, for example, cancer, we noticed that non-coding RNAs are something that could also be applied in neuropathic pain. So we decided to look at microRNAs, which are tiny molecules in our cells that act as molecular switches. If we remember our central dogma of molecular biology, these microRNAs, tiny molecules, are acting actually on mRNA, and they have power to turn on and off our genes. And microRNAs are also very useful for clinics because they can be used as diagnostic markers, predictive markers, and even therapeutic agents. Considering all this, we designed our study in order to look for these particular signatures in skin biopsy of our patients. Skin biopsy is one of the most widely used tools to diagnose our patients. Because it allows us to look at the morphology of small nerve fibers that are transmitting pain. We therefore extracted RNA only from the part that we considered as a microenvironment of these tiny fibers, proceeded with microRNA profiling, and performed the bioinformatic analysis. Very early on in our study, we understood that, that there are a lot of discrepancies in the literature when it comes to microRNA analysis. Therefore, we invested some time in creating analysis pipeline that we also published. Only then we were able to analyze our data. As a first patient group, we looked at a very homogeneous patient group that we called painful sodium channelopathy patients because they suffered from neuropathic pain associated with pathogenic mutations in sodium channel gene. All our patients, in this cohort of patients, we discovered 4 downregulated microRNAs that then we associated through adding the functional study as microRNAs that are regulating pain transduction and transmission. In the second cohort, we looked at patients with idiopathic small fiber neuropathy. These patients were characterized by severe denervation of these tiny pain fibers. And in this second cohort of patients, we identified 2 different microRNAs that we associated with pathways that are axon regeneration and maintains. At the end, during my PhD journey, I learned that in order to comprehensively understand and explore neuropathic pain, we must apply multidisciplinary approaches. I learned also that the genetic background can contribute in various ways and that we should not overlook it. I also learned that microRNA indeed can be really good disease predictors, biomarkers, and I hope therapeutic targets. I also was very lucky to land in a group that did translational science. So I learned to appreciate that bridging the gap between basic science and clinical practice is crucial for treatment development. And also I learned that integration of different research methodologies is essential for advancing our understanding of neuropathic pain. And with this I end, and I arrive to acknowledgements. I'm very fortunate to have a lot of people on the list to say thanks to, but it's too long for this session, so I will just say thank you to all of you for contributing with your skills and background to these works and to my journey. And I thank to my supervisors, Professor Karin Faber and Giuseppe Lauria, for their guidance. Thank you all for listening, and thank you.
Thank you very much for your very clear and beautifully illustrated presentation.
Thank you.
Your parents might sit down now, otherwise they get painful feet and might hit the topic, but it's not a solution for that. We will start with the questions of the opponents. There are 2 people who will not say anything, because that's Professor Lauria and Professor Faber. They have questioned you enough, I guess, so we leave it to the others. So the opposition will be opened by Professor Joosten. He is the chair of the assessment committee, and he holds a chair in experimental anesthesiology and pain management of Maastricht University. The floor is yours.
Dear candidate, first of all, I would like to congratulate you with, I think it's very important and very good work, and include also the promotion team with this. Small fiber neuropathy is a complex disease, and for pain researchers like myself, very difficult to understand. The golden standard is the intraepidermal nerve fiber density, Yeah, a reduced number of fibers results in more pain. I never understood that. So how come? But I think your research and the interaction you showed in the epidermis between the keratinocytes and the nociceptive endings, and your elegant molecular analysis has made me understand, at least to some extent, what is going on. So that was really great.
Thank you.
Yeah, but then having and trying to understand, I come to my question, which is a very open question. My first question is a very open question. Is if you then look to chapters 4 and 5, and the figures 4 on page 148 and figure 4 of chapter 6 on page 198, it's extremely complicated. In this field, I thought 3 years ago, when the Nobel Prize was given for the transient receptor potential vanillin family, okay, that's a major breakthrough. These are the key molecules. But then having a look at what you present, it becomes so complicated. So the question is now, where are we? And why does nature make such a complicated network of microRNAs and molecules in a relatively, in my opinion, maybe a relatively simple process? Could you elaborate? Tell me a little bit more about that.
Well, thank you, highly esteemed opponent. Thank you very much for this comment and your question. It is a tough one, I must admit. I don't know how to answer that based on data, because I don't have it. I don't know how to why these pathways are so complex. But I do know that, you know, if we look at pain and what it is, it's a complex, it is a complex situation. I don't know how to call it. So I believe that, you know, the interaction of all these cells are very important. And if we also remember that cell body of our C fibers is very far away from where they are in the skin, something needs to be there to support their regeneration that occurs frequently. So I think that, you know, if we need to have these mechanisms that are working so in this distant tissue, that they need to be as complex. I don't know, but maybe they are not. I don't know how to.
Yeah, when I talk to students or try to explain, I make it maybe too simple, but then the TRPV1 is temperature sensitive in a particular range, so you have a set of nociceptive endings.
Sure.
For that, you have other set with the TRPV2. You have, and you talked also about the TRPA1 and the TRPM, the mental for the cold. So that's a key molecule, and probably attached to that are general processes. Do you agree, or is there also a specificity with each set of nociceptors,
well,
which are identified by the Nobel Prize winners? So just to emphasize that must be a key in finding.
Yeah, well, I agree that it is a key finding, but I mean, we didn't look at a specific receptor. We wanted to understand what is happening in this tissue, because, I mean, these people felt pain there. So, and as you said, there were no fibers that transmit pain. So something has to happen in order for them to feel pain. So, yeah, I think this is very complex question. And I don't know how to really answer you, because
that's no problem. It's just more general than a general. So let's then maybe a little bit more specific. If you talk about small fiber neuropathy, a key event is neuropathic pain, is central sensitization, and you want to test your nanotechnology in some kind of a drug testing in an in vitro assay. But you did not include central sensitization because I think, I would say, the proof of the pudding is in the eating. And finally, you want to see that central sensitization, which occurs in the spinal cord.
True.
What's the first? There's the gate on-off and sending the signal to the brain. So you did not include that, or do you think we should include that in the next step? And how should we include that in the next step?
Well, thank you for also this question. I think, yes, it would be perfect if we could include all the aspects in order to study what's happening to our drug. So I think that, fortunately, for today, the best models to test drugs, even the nanotechnology, are animal models, because they then have all the parts we need to understand where this drug is acting and how and what side effects can occur. In vitro models, it's very hard to have all these parts and to know what will happen then. But we also do have in silico. We have computer science now that is a lot. Also, we in BESTA we have really good mind lab that is a bioinformatic group, and I believe that maybe they could predict somehow what is happening in interaction, which kind of interactions are happening if we are introducing a specific drug. I don't know if that can.
Don't you think that the finals are what the step in between the in vitro and the clinical application is an animal model where you then really test the pain behavior, but also electrophysiologically,
sure, yes.
say the output of the cells of the pain neurons in the spinal cord in relation to the effect of your nano approach?
Yes, I agree.
Yeah?
That electrophysiology.
Okay, we agree. We do have such an application and setup in the lab. Anyway, a next question, if I may. A very short question. Okay, let's make it short. Yeah, you want to address the nanotechnology to modulate the microRNA 26B5P. And if you then go back to the figures, you see that this microRNA, let's say, regulates a lot of effects. Don't you think, if you will end up in a possible therapy, that you will have a lot of side effects?
Yes, I do, and I agree with that. But I think that nanotechnology is also the group that we are working with. They're really smart, so we can actually tag our vector that is carrying our microRNAs to go into direct target. For now, we are working in order to direct them to specific cells that are keratinocytes, because from our research, we believe that they somehow influence the neuronal growth, but also that could be a mistake. So we have to test that first, and then hopefully we can even target specific gene targets to drive these nanotechnologies, but I'm not an expert in that, so that's something I hope to learn in future. Okay,
yeah, I think, and as you indicated in the impact paragraph, you will try to find the super molecule, so maybe this will be the super molecule. I will give the word back to the prorector.
Thank you.
Always, we are creative. So thank you, Professor Joosten, for your questions. We will now continue with the next opponent, but I see one should be online who isn't there, so we will change that. So I first give the floor to Dr. Menicella. She's an associate professor of the Institute of Northwestern University Feinberg Medical School, Chicago. Nice to have you here, even if it's a long distance and very early morning for you. Welcome, we much appreciate you being here. So the floor is yours.
Thank you. This is my pleasure. And I will address a question. Dear candidate, congratulations on your beautiful work on this complex and relevant health issue, which is small fiber neuropathy and neuropathic pain. And I'm really intrigued about your approach, your exciting line of research, which investigates the communication between cell in the skin and cells that carry pain, the nociceptor. And for the first question, I would like you to expand on the correlation between the different sensory phenotype, as you mentioned, the different way the patient describe their pain in this large population of patients with small fiber neuropathy, and your microRNA. So on the translational perspective, how you think we can use these microRNA practically with this expression in the skin as a biomarker for identify and also monitor longitudinally different sensory phenotype in this patient population? And are they able to distinguish pain and non-pain for small fiber neuropathy?
Question. It is a very nice question because it's actually what I was curious to understand, because as I said, I saw a lot of patients in the clinics with our clinicians, and I understood that, you know, even if they have the same disease, they have different symptoms. So we hoped with this microRNA approach that we can somehow stratify them better in order to then drive maybe a therapeutic choice, because maybe there are therapies that they are already taking and responding well, because we know that there are some patients that do respond well to therapies, but we don't know how to select them, how to, because the biomarkers are not specific yet. So to answer your question, I hope yes. I think that we still have to expand on our population that we studied, because we didn't have a lot of patients, and all these approaches are also very expensive. But with our data was robust because we applied also different kind of statistical approaches in order to make sure that this data is robust. But yeah, and I really hope, and to answer your question actually more specifically, I think yes, that these microRNAs can serve as, in future, of course, as good biomarkers for this kind of patients, because among our patients, populations, for both studies, we had patients with that described their pain differently. But they still had these microRNAs, the expression of these microRNAs downregulated in both cases. So even if they complained of different symptoms, they did somehow, they were a homogeneous group when we looked at microRNAs. So I hope, yes, that in the future these microRNAs could serve as biomarkers. But as I said, I think we have to, expand and test more patients and probably do validation studies that I would be really happy to do.
Yeah, this is a very important work, and I find a really good strategy because of the accessibility of the skin biopsy, as you know. I do have another question, and I see if you can help me to understand, in a sense of the skin is a very complex biological system, and so there are many different cell types that are housed either in the dermis or in epidermis. And just looking at the epidermis, at the more superficial layer, how are you going to validate your result to assign the different microRNA profiles to distinct cell type population in the epidermis or in the dermis?
Thank you also for this question. We decided to look at epidermis because there is where degeneration occurs, and that was our first drive to design this study. But yes, I agree with you that all the other layers probably are equally important, and that probably the same microRNAs are behaving differently in all these different layers of the skin, but also in different cells. So we kind of looked at a microenvironment. That's the word I really like, because I think that all these cells that are in the epidermis, or that we looked for now at, are equally important, and they are creating the environment for our nerve fibers to regenerate, to grow, to feel different sensations. So that was something that I wanted to understand, how this microenvironment is behaving, and if it's hostile in some way for these fibers, and that's why these patients feel these symptoms. but yes, there is more to it for sure. And I think that it would be really nice to do maybe another approach like single cell approach or attack sequencing or different kind of sequencing in order to understand which cells are contributing in which way and how can we then design our therapies and also diagnostic tests.
Yes, yes, I share your fascination for this little layer in the skin, which is so complex indeed. I'm satisfied with your answer. I do have another question if I have time, though. And now that you gain so much understanding on the molecular mechanism in these small fiber neuropathies, I have a question. Do you think it is the regeneration of or the degeneration of these fibers that it is the culprit, the one causing the pain?
This is a fantastic question that I don't have an answer to, not the evidence-based one, and I would really love that I do have. But we know that there are works showing that regeneration actually can cause equally pain because the fibers that do regenerate somehow change their phenotype, and that is something that I would like to investigate in some kind of a model. But for now, I really don't know what answer to give you. But it's a really valid question, and I hope that we will have an answer in future.
Thank you very much, Dr. Manichella. Are you happy with the answers?
I am very satisfying with the answers.
Thank you very much. Then the opposition will be continued by... yes, there she is again. Fantastic timing, Professor Sommer. She has a chair in neurology at the Universitätsklinikum Würzburg. Thank you very much for being here, and the floor is yours.
Thank you, and thank you, dear candidate, for this really very nice presentation and for your good work. I'm very pleased to hear that as a basic scientist, you also tried to understand the patients. And therefore, my first question is about the patients. Those people with congenital insensitivity to pain, do you have any idea how they can grow up? Can they learn what pain is? Do they have compensatory strategies? I would be very interested to hear what you know about this.
Thank you for joining and also for your question. Honestly, I don't have any idea of if these people can understand what pain is. I don't even know if they no. I don't know if they can understand the pain. But I think that I don't know. It is a very difficult question. Honestly, I only was introduced to one of these cases, and I was fascinated by this case because they're very rare and very extreme. But I only know that that people that suffer from congenital insensitivity to pain, that their lives are very difficult because their interaction with the environment it can be always dangerous because they don't have this sensation of pain. But honestly, I don't know if they can in some way feel pain. Probably
okay, let me ask.
Depend on the type of mutation they have also and the extent of damage, and depends also maybe which kind of the protein that is made. If there is some residues of wild type protein that can somehow give them some sensation, I don't know. Probably it could be also due to the type of the mutation they have.
Okay, let me ask you something more in the science area. So I'm also very intrigued by your work on TRPA1. And in one paper, your group used a gene-wise aggregation analysis Can you explain in simple words what this technique does?
It is actually, generally, when you do a genetic analysis, you look at a single gene and analyze variants in a gene in a patient. But this analysis is more a statistical one, because what we did is we looked at population, and if there is a gene that is enriched by variants in considering all patients, not just a single patient. So it is more like a statistical analysis. So what we learned is that this gene was enriched with mutations, and in a particular domain that is very important for the function of this channel. And then we also compared it to healthy individuals, and we saw that this gene is not as enriched with mutations in a healthy population. So it was like statistical analysis of variants in considering entire population of patients.
Okay, thank you very much. And just one follow-up question on the TRPA1 story. Why do you think are the phenotypes of the variants so different? Or at least there are some quite different ones.
Thank you. This is a very valid question, and we actually are wondering the same. So we are trying to set up collaborations with electrophysiological groups in order to try to assess these mutations also from electrophysiological point of view. So maybe that data will give us some answer on how on correlations with different phenotypes in patients, and help us understand why there is so much heterogeneity.
Okay, thank you very much.
And also, sorry, I wanted to say that this analysis provided us a risk. So it's not causative. These variants are not causative. So they are just contributing to overall risk for developing neuropathic pain. So probably others factors like other mutations or other epigenetic factors are contributing to overall phenotype.
Okay, thank you. That's a very clear explanation. So I will move back to the chairman.
Thank you.
We still have time for another question if you want. But if you are happy, then you are happy.
I'm happy for the moment, and give the other opponents a chance. Thank you.
Thank you very much. And then the opposition will be continued by Professor Linden. He is a member of the assessment committee. He has a chair in translational neurosciences of this university, and he is also secretary of this committee. Quite a number of functions on one day. So the floor is yours.
Dear candidate, also from me, congratulations to you and to your promotion team for this very interesting, very detailed work in translational neuroscience of pain, and also very nice illustrations you provided. And you also have nice quotes. I quote from your discussion, page 220, quote from C.S. Lewis: "Pain insists upon being attended to. God whispers to us in our pleasures, speaks in our consciences, but shouts in our pains. It is his megaphone to rouse a deaf world." Why do you like this quote?
Highly esteemed opponent, thank you for your comments and also for your question. I read this quote actually in one of the papers of Professor Waxman. He entitled his review with this sentence that pain is a God's megaphone, and that we need to find ways in order to silence it. So I actually liked that part of this quotation. And I also do agree that pain is very important. As I said also in my presentation, it protects, in a way, during this PhD journey, I learned that it's not just terrible experience. It also warns us about different dangers and tells us if there is something wrong with us. So actually, it's a positive in some ways. And so I like that quote for these reasons.
And certainly it would imply that the attention aspect is a central process, and central being in this case related at least partly or strongly to the CNS and more specifically to the brain, which of course you can image during a pain experience. And you have on figure 2 and figure 2 page 5 you have the battery of diagnostic procedures. And so what's the role of functional imaging to look at brain correlates during in pain patients? How can it help you with diagnosis or understanding mechanism?
Thank you for that question. Honestly, I am, I was not working hands-on this aspect, but I know that psychologists, for example, in our group, she's studying different patterns in the brain that react when in different patients, and trying to understand if some psychological aspects, some psychological trademarks of these patients are associated with their experiencing pain. But honestly,
that's fine. I mean, you also mention treatments. So what I mean, so if I read page 6, first-line treatments, tricyclic antidepressants, anticonvulsants, serotonin, noradrenaline reuptake inhibitors, and others. So how do they alleviate pain?
All these
peripheral or central action or combination of those?
All these different drugs can act centrally and only centrally. Some of them are acting on also peripheral nervous system, but and yes, in a way they
I mean, might some of them actually sort of affect the attention to pain rather than the bottom-up processes?
Yes, that could be. Yes, I believe that they are acting on also attention of pain and how.
In your conversation with patients, did that come up? Patients trying to sort of use strategies to reduce the attention to the pain, or is it so overwhelming that that's not really?
Well, there are patients that react well also on if they are more engaged in an environment. But there are also patients that because of their pain they are not engaging too much with other people. So I think that attention has a lot of that is very important but also it's very subjective, I think. In a way, because for some people something works better, and honestly, not being a clinician, these things are not something that I'm hands-on every day. Also the therapies, I only know that they don't work, and that but I also do know that there are some patients that respond well to, for example, psychotherapy. Some of them are undergoing nerve stimulation techniques as well. Some of them are really good candidates and feel very well after that. We have examples in our hospital, but those are things that that are not focus of my research. So I'm
not sure. Perhaps they are, because you're looking at genetic changes. In your case, mainly microRNA. If you you looked at in skin biopsies, that makes perfect sense. Where would you sample if you had a chance to do the similar analysis in some novel technology in other parts of the processing chain? Where would you look? Where would you expect?
I would like to have, for example, liquor of these patients, spinal tap. That would be very interesting, I think, to look at microRNAs there or different proteins. I think it would be relevant.
Yes, very interesting. I'd like to move on to your theses, to your Stellingen propositions, and I think one of your paronyms I'd like to invite to read proposition 5.
Proposition number 5. The combination of molecular stratification endotyping and clinical phenotyping will revolutionize neuropathic pain management in the future.
Yes, thank you. And I think this also relates to your impact paragraph. So, well, can you expand on this a little bit?
Yes, in our group, we are working really hard to understand, to look at pain, not only from a clinical point of view, but also from molecular one, hoping to find different patterns and different molecular signatures that we call endotypes. Because what we saw from also clinical trials that have been done, that they are failing, even if we are treating patients with, sodium channel blockers, but they're still failing. So maybe because the patient selection is not as good, and maybe if we would, because there was a clinical trial on a couple of patients with sodium channel variants as well, and from the group, from our colleagues from Maastricht, and they saw, for example, that there are patients with sodium channel variants that do not respond well on lacosamide, which is a drug that should act there. Maybe these patients had something else that contributed to this overall non-responsiveness. But microRNAs, for example, if we take an example from breast cancer therapy, where microRNAs were co-administered with a drug for women that were non-responders, they started to respond to the therapy. So, we are looking at microRNAs from different angles and trying to understand if they can somehow improve the rate of responders, if we can somehow improve the rate of responders by modulating the existing therapies, or by selecting better patient cohorts that we will then treat with specific drug. For example, our patients with TRPA1 mutations, there were studies that carbamazepine works well in this kind of patients. So maybe that could be a next hint for clinicians to, if you know that this patient has this kind of mutation, maybe that would be the first drug of choice. I don't know, I really hope in future that is gonna be like that.
So presumably this use of existing drugs and then stratifying patients is you get faster gains, faster successes than actually having to develop new compounds that target the microRNAs, which may, as Professor Josten mentioned, also have, because of complexity, lots of side effects. And might you also use the microRNAs as early biomarkers of treatment?
They can be predictive biomarkers, but I was in this context, I was thinking about more to predict drug responsiveness rather than be early biomarker of disease. I think that in the field of pain, we still have to do more of this research, and probably some other models would work better than idiopathic small fiber neuropathy. Maybe in patients with diabetic neuropathy or chemotherapy-induced neuropathy, we could track the development of disease and then try to understand if there are predictive biomarkers that we can then use in future.
Thank you. I'm very satisfied with your questions. I give the word back to the pro-rector.
Thank you.
Okay, thank you very much. And now I give the floor to Dr. Hoeimakers with an expertise in neurology from Desi Institute. Welcome.
Thank you. Dear candidates, also I would like to congratulate you on this wonderful thesis, and also the congratulations for your supervising team. As a clinician, I'm really grateful for your work because important steps have been taken towards better treatment options for my patients. And of course, I also want to exchange thoughts about some parts of your thesis. And I first want to go to chapter 2, the chapter about congenital insensitivity of pain, which is a very interesting condition to study, to understand the pathophysiology of pain. And I have the question about the skin biopsy you have taken, because I see a lot of patients with painful small fiber neuropathy, and those patients have a decreased intraepidermal nerve fiber density in the skin biopsy. And they always ask me, why is it reduced and I feel the pain? That's what also Professor Jooste already asked himself. But I saw that also your patients with insensitivity to pain has a decreased intraepidermal nerve fiber density. Can you explain why both the painful and painless neuropathy patients have a decreased intraepidermal nerve fiber density?
Esteemed opponent, thank you for your comments and also for your question. I think that's a relevant question, and honestly, the best answer I could think of, even if it's not evidence-based, it's something that that I come up with now, is that probably because of the unfunctional sodium channels that are very important for pain transmission and overall function of these nerves. If they are not functioning and not there, maybe these nerves are just not regenerating, and eventually they are going towards degeneration. So probably it's different mechanisms that are inducing this kind of degeneration than the ones in patients that do feel pain.
Mm-hmm. Yeah, because in our patients with the SCN9A mutations with the painful small fiber neuropathy, there is a gain of function and you see an increased sodium influx, and then the sodium-calcium exchanger can change. But I think that will be different than in the painless patients.
Exactly, yes, I agree. And since there is no this exchange interaction with the environment, probably these fibers feel that they don't have any need. So maybe also that could be an explanation. But it's something that it would be really nice to understand. I agree with you.
Yeah, and it's still a question why all patients feel pain without the nerves, but
yes, true.
Then I want to go to chapter 3. That's the chapter about the TRPA1 variants. And yeah, one of the aims of your thesis is to achieve a more precise patient stratification by identifying objective biomarkers. But of course, you started with the phenotype, and yeah, that's my work as a clinician. But I got a bit confused about that, because on the one side, there is a group of patients with neuropathic pain. And as you already told in your layman's talk, that is based on evidence of a lesion or disease of the somatosensory nervous system. But on the other side, there's a group of patients with nociplastic pain, including the widespread chronic pain and the fibromyalgia. But when I look at your table 2, at page 51 and 52, also in the nociplastic pain group, reduced sural snap amplitudes were found, and also abnormal skin biopsy have been found. So why did you stratify those patients as having nociplastic pain?
Thank you for that question. Honestly, I didn't. Clinicians did that part of the job.
Yeah.
But I think that those are all neuropathic patients, but with also nociplastic component in a way. And I think that it would be really nice because all these patients come from different centers as well. So I think also that could introduce a different interpretation because of the patient's symptoms. So I think that it's a mix of different things that happened in there. But I think that the symptoms still were the widespread pain. They felt pain everywhere. So we managed to somehow correlate the phenotype that they had with the mutations. And as I said, these mutations we don't consider yet as functional as pathogenic ones. Mm-hmm. So they only represent a risk. So maybe everything else that happens in these different kind of patients, also maybe environment from which they come from, epigenetics, everything that could influence different other different genes, and then again variants, and then again this complexity in the phenotypes. But that's something that me as a biologist, it's really hard for me also to understand.
Yeah, I can imagine. That's the same as for me. If the microRNA studies are quite hard to understand. So I understand that. And in the end, irrespective of the clinical diagnosis, you have found the TRPA1 variants, you already told. My question is my clinical assessment as a medical doctor still necessary? Or can I say to the patient, well, I don't matter, let's go to the lab, and they can perform genetic analysis, and then we find directly a target for treatment. How do you think about that?
I think that your job is absolutely necessary, and I learned that from interacting with a lot of clinicians. Also for us in the lab, it's very important to have your input when we are designing our studies. And but without you, how could we know which patients to put in which cohort and who to analyze it with which approach? And again, how to design our studies. And in clinics, I mean, patients, I've seen you, other clinicians working with these patients, and I think it's really, really hard job, and I admire you a lot. So you are absolutely necessary. And I think, even if this data is promising, that we still need some time until it comes to application in the clinics. And then you would need to decide which kind of test you would do to which patient, because they are very expensive. So, I think your job is safe.
Well, I'm happy to hear that you are happy also with our work. But, well, in a general discussion, you mentioned that patients with a mixed phenotype can be excluded from clinical trials because, the phenotype is not that clear. But do you think that then it's better to use the genotype as an inclusion criterion for a clinical trial, or?
Yes, I think that would be very helpful to include also from the study, your study that we know, at least things that we know to go targeted to test for those things, to these genes and these variants that we know what they are causing for now. Yes, I think it would be very helpful.
Yeah, in all studies there was some variants of uncertain clinical significance. Do you think that's okay to include them, or do you think it's better to only include pathogenic mutations?
Well, I am really, in a way, stubborn. So I like to, I would use only the ones that are provenly pathogenic, because with the VUS we don't know if they are and how relevant they are. So maybe it would be a cost that is not necessary. So I would for now focus on the pathogenic mutations.
Yeah, that would really be ideal, but maybe then it's harder to get some patients, but when we work together, there are maybe more patients, so that's a good one. And in your impact paragraph, you also have a new suggestion for organizing clinical trials, and then it's not the phenotype, it's not the genotype, but it are the microRNA biomarkers. If you can choose, is that even better than using the genotype to include patients in your clinical trial?
Well, I'm biased in answering to this, but I believe that microRNAs are really powerful in a way that they can, we call them mirrors of disease. So they can, in a way, mirror what is happening to entire organism. That is what we believe they are. So, but I would really like to have more proof in order to say that really. Strongly. But yes, I believe and I hope that they can be helpful in this way in order to stratify patients better. Yes.
Yeah. And then I can tell my patients, well, my colleagues found some great things. They can find biomarkers in your skin biopsy. So come to my study. But how long does it take to analyze a skin biopsy for the microRNA?
It is actually not as difficult. The extraction is like a protocol that is 1.5 hours, RNA extraction, and then the post-processing is very easy qPCR that every lab does. So it's not as difficult.
So I have an answer in 1 day?
Yes. No, 2 days. 2 days.
You may answer the question.
It takes about 2 days for the wet part, and then depends on analysis. How long does it take? But we have now pipelines that are ready, so we hope it's very fast, fairly fast.
Sounds good. Thank you very much.
Thank you. Well, you probably noticed that Patel came in. She hit the floor with the baton. It's still in one piece, so your defense has ended. I would like to ask you to sit here and wait together with your company, and we will withdraw and talk about the quality of your thesis and your defense. So
Okay.
we will return shortly.
Thank you
Yeah.
very much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Dear Mirna Anđelić, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Faber is authorized to confer upon you this academic distinction in accordance with the Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Mirna Anđelić, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
Thank you.
So good afternoon, and dear colleagues, dear, actually not candidate anymore, so dear Mirna, congratulations on your defense and presentation, and actually on the work you've done during these years. Actually, I've been in this position some other times in my life, but this is a very special day because actually we are celebrating not only a brilliant new PhD graduate, but also the work that between Maastricht and Milan we've done in the last 20 years. So this is another exquisite result of a very long story to which we have been committed in order to provide patients with a new, better knowledge of neuropathic pain, but also to concretely advance the generation of new scientists, joining and putting together and mixing medical scientists and basic scientists, which is very, very important. Actually, Mirna is a brilliant, passionate, diligent, young scientist, never satisfied with the first answer to a question, and able also to say, I don't know, which I think is very important in life in general, but in science as much. She's been part of a group of 13 young graduates which have been selected within the Marie Curie project granted under the European Commission, which I had the honor to coordinate. Actually, that project has been designed to actually to foster the dialogue between clinicians and scientists. And I think that the work that Mirna today has presented witnesses the achievement of this journey. Well, when Mirna came to Milan after being selected, I might say at first I was a little bit worried because you know, because of the charm of Milan, the fashion, the exciting life, the attraction of this of our city at that time, so attractive. But actually, I was wrong. Mirna was just getting her bearings, was analyzing the context, and after a few months, I remember it very well. On a Friday night, it was about 7:30. I received a text, okay, with a photo on my WhatsApp saying, "Prof, the cells in the culture are growing wonderfully." And actually, at that time, I realized that Mirna had become really a young researcher in every sense, even aesthetically, okay, with hair tied up and held with a pencil as a symbol. And actually, this is what this has started, and actually had the privilege to follow her during the years. I saw her bloom, facing the challenges of science, of the complex experiments, and also the fear of failing, but with the determination of a person who wants to succeed. Always, I must say, with kindness, never arrogance, and fairness in the relationship with the colleagues. And in this way, I believe, she found her space along with her identity through the strength of the knowledge she was nurturing. So for me and all my collaborators, it has been really a pleasure to have Mirna in the lab. I know that she has the right ambition to grow and expand her cultural and scientific horizon, and I wish her to achieve the next goals while always keeping in mind that in Milan, in our Carlo Beste Institute, she has her home, she has her alma mater, always. So congratulations again.
Thank you very much, Prof.
Dear Dr. Anđelić, also on behalf of this university, congratulations with this result. In this historic place, you wouldn't recognize this, but it was actually the chapel of a monastery. They kind of rebuilt it, and it has a kind of an elaborate history. Napoleon used it as a stable for his horses and things like that. So it has its ups and downs, and today was definitely an up. You did a wonderful presentation, and you had a wonderful defense, so thank you for that. We'll shortly conclude this session, but before we do that, I want to point out that we first take a picture in front of these screens so that your other opponents online are there as well, and they can congratulate you online as well, so you get to talk to them. That's probably better. And then we take the final pictures on the big staircase when you come in. There's a big staircase, so the pedal will guide us there, and then we will have drinks, which is always important in Maastricht. So thank you very much. I close this session.
Okay.
Recording.
So, are we excused?